Yeah, and please briefly acknowledge also our disclaimer displayed here on this slide.
Thank you for the nice introduction and welcome to our webinar today. My name is Jenja Schlegel and together with my colleague Sarah Rabati, we will give this webinar about spatial biology at its best. In the first part, I will explain a little bit about our complete workflow solution that we have to advance your immuno-oncology research. And in the second part of the webinar, my colleague Sarah will talk about spatial biology applications and, for example, how to identify new CAR-T targets. And I would also like to mention that this webinar is part of a webinar series, Explore the Full Potential of Spatial Biology in Immuno-Oncology Research. The next webinar will be held on the 10th of May by Amy Jablonski, and she will tell you about predefined antibody panels and how to use them for the tumor microenvironment phenotyping of solid tumors. On May 17th, Thelma Santos will talk about our data analysis software, and she will guide you through the analysis of a spatial biology data set. We will close our webinar series on the 24th of May with Egle Quadreite, who will talk about mononuclear phagocytes in tissue from inflammation to cancer. And she will present very new data and exciting new research about how myeloid cells play an important role in our immune system. Okay, after this introduction, I would now start with our webinar Spatial Biology at its best. So to start our webinar, I would like to define the term spatial biology and what it means to us. And here you can see a tissue section of a colorectal cancer, which was stained with a lot of different markers. Here visualized are 8 different ones, and you can already see how many information you get out of this. So spatial biology for us means that you now can provide the information on the expression levels of not only 3 or 4, but hundreds of protein markers on individual cells within the spatial context of your tissue. And it's important to not only look at the tissue and look at the different markers, but also to analyze the tissue. And so usually here the first step is the segmentation, and with this you can then now give individual cells a unique ID, and you can really start to do a very in-depth analysis of your tissue. Why would we actually need so many markers on one sample? Why do we need to look at hundreds of different markers on our tissue sample? So in the end, why do we need spatial biology? And if you look down here in the right corner at the cartoon, this is derived from an actual data set. You can see the tumor microenvironment, and you can also already see how heterogeneous it is. And if you look at the different cell types and all the different cell types that the tumor microenvironment consists of, you see how many different markers you already need to just characterize and look at your tumor microenvironment in detail. So already to just characterize the tumor microenvironment, you do need hundreds of markers to really identify all the cells and all the subtypes of your cells. So our solution for spatial biology is the MACSima imaging platform with the MACSima imaging cyclic staining technology. The mixed technology works in 3 different steps. In a first step, we stain the sample with up to 3 primary conjugated antibodies, which are attached to 3 different fluorochromes. In a second step, we then image these 3 different antibodies within the machine. And in a third step, we erase them again. So there we either erase the antibodies or the fluorochromes. And since this erasure step is so gentle onto the cells, we can repeat the cycle over and over again so that we basically can stain an unlimited number of markers on the samples. And this is here an example then how it looks like. And what I would like to mention here really is that we do not offer only the instrument itself, but a complete workflow solution, so the complete Maxima imaging platform. And the first part of this platform is our instrument device. And this device is a fully automated sample processing and imaging machine, which is called the Maxima imaging system. And it consists out of 2 computers, a liquid handling system, an ultra precise stage, which is always going where it's needed. At first to the liquid handling system, where the antibody solution is taken up and putting onto the sample. And then the stage is driving to the state-of-the-art microscopes and cameras, and this microscope consists out of 2x objectives to at first take an overview of your image. There you can then select the region of interest, and then you have 2 20x objectives that give you in the end a single cell resolution of your tissue samples or your cells. And all of this is happening fully automated. So you put in your antibody panel and as well as your samples, you press play, and then you can go on with your day. Our second aspect of the Maxima imaging platform are the sample carriers, and these sample carriers are specifically designed for all kinds of fixed samples. And so, for example, if you have your tissue already on a standard microscope slide, for example, your FFPE tissue, then you can just use one of our Maxwell imaging frames, and these ones you click on top on your standard microscope slide, and you don't have to move or change your tissue sample. We offer these Maxwell imaging frames in different sizes, and Maxwell 1, Maxwell 2, and Maxwell 4. So depending on the size of your sample, you can choose the different sizes. These samples then just go into the Maxima machine. We also offer a solution for adherent cells. So if you have adherent cell culture, you can culture your cells in our Maxwell 24 imaging plates. And then also here, you culture your cells there, and then you just place them into the Maxima machine. And with this, we are already at our third pillar of our Maxima imaging platform, and this is our broad portfolio of pre-tested antibodies. So we offer over 500 different antibodies that are pre-tested for our mixed technology. We offer these antibodies in 3 different formats. The first one are the dissolved antibodies. So our pre-tested antibodies for the mixed technology come in these off-the-shelf vials, which offers you a high flexibility to design your own antibody panel, and then you can pipette the different antibodies in your 96 well plate like you would like to have it. However, we also offer some rare screen plates, and here we have some predefined panels. These are ready-to-use dried antibody panels where we used standardized markers, for example, to describe the tumor microenvironment in solid tumors. And this offers you reliable and reproducible data since you don't have to do the pipetting yourself. Therefore, you also decrease the error rate. Also, it is convenient and time-saving since you don't have to think about your panel design and you don't have to pipette them. And also, it is very efficient. To add to all of these advantages, also the flexibility, we also offer some customizable panels. So you here can choose which antibodies you would like to have, and then we will design you your rare screen plate, and we will send you this dried antibody panel plate that you can then use. With this, I would like to already come to our last and fourth pillar of the Maxima imaging platform, and this is also one of our highlights, which is the MaxIQ View software analysis. Our MaxIQ View analysis was tailor-made for these kind of data sets, so it's one of the best softwares on the market. You can easily visualize all of these different markers. It has an easy and fast segmentation process. You can do different gating strategies. You can do distant maps, so you can see how far away are your immune cells from your tumor. We have solutions for clustering and dimension reduction, so you can do t-SNE plots on UMaps. We have a really easy to use workflow editor so that you can build up your analysis pipeline and then use the same analysis pipeline for different experiments, or you can change it really easily in this workflow editor. And of course, there is the option for a nice data visualization and the export of your files. With this, I now presented all 4 pillars of our Maxima imaging platform and presented our complete workflow solution. So we offer the fully automated instrument. In addition to this, the nice sample carriers and this broad portfolio of antibodies that are pre-tested for our mix technology. And to end it, we have our highlight, the MaxIQ View software, which is a great data analysis software. So all of this together comes in one package with the Maxima imaging platform. With it, I would now like to hand over to my colleague Sarah, who will talk to you about spatial biology applications. Thank you.
Thank you, Genia, for the introduction. Good afternoon, everyone, and welcome from my side as well. As we just heard, Maxima Imaging Platform allows us to perform and analyze spatial biology. And for that, I would like to take you to the next part, which is the application of spatial biology and Maxima Imaging Platform. In fact, the application of spatial biology, same for Maxima Imaging Platform, is quite wide, ranging from identification of new biomarkers to drug discovery and cancer research. And in fact, today I would like to talk to you about one specific application on drug target discovery and validation. The application that I'm going to talk about today in the next few slides is based on this paper published in Nature Communication. The aim of the study was to target PDAC or pancreatic adenocarcinoma with CAR-T immunotherapy, and for that they identify new target candidates. This is a work of our R&D team at Miltenyi Biotec, done by Dr. Daniel Shefa. You can download this paper from this link or by simply scan the QR code. This study has 2 main parts. The first part is to identify the tumor-specific and safe target candidates, and then second part is to generate those target candidates and evaluate them further in vivo, in vitro, and in silico. The study started with patient-derived xenograft models that were screened and analyzed for 371 markers using flow cytometry analysis. This was done together with in silico analysis of RNA and protein extraction databases. The next step, 97 markers were chosen, and they were tested for their spatial distribution in human tissue using Maxima imaging platform. And then this was followed with a additional sample verification of chosen target candidates using flow cytometry. And then finally, the selected target candidates were validated for the off-target effects using Maxima imaging platform in healthy human tissue. Here, the outcome of this first part was 3 target candidates that were generated in the next step and validated in vitro and vivo, with the final validation of those targets using Maxima imaging platform in the full picture of tumor tissue. This is the second part of the workflow where Maxima imaging platform is used. What we see here is quite unique. We are looking at 100 of markers in a single PDAC tissue sections. In blue, we see DAPI, in red, EpCAM, in green, the marker of the interest. And what we see here is the spatial distribution of each marker, and this allows us to distinguish between tumor-specific markers and non-tumor-specific markers. And this information is something that is lost using single-cell approaches such as flow cytometry or RNA-DNA sequencing. But Maxima imaging platform allows us to perform spatial biology, looking at the target candidates within the native tumor tissue. Here is a closer look into multiplex analysis of PDAC tissue. In this example, CD95 appears to be present everywhere except in the tumor cells, which is labeled by cytokeratin. We know that cytokeratin is a marker of epithelial cells and can be expressed also in healthy cells, and only by spatial biology analysis and morphological assessment we can only distinguish between healthy and tumor cells. In the case of CD318 and CLA, they are both co-expressing with cytokeratin, but with much higher level of expression in CLA. So far, the selected target candidates showed high tumor specificity and had the highest safety ranking in silico analysis. But now we want to look at this evaluation of the safety of the candidates in situ. And for that, Maxima imaging platform was used to ensure that the selected surface markers specifically and only targeted tumor cells without harming normal healthy cells. Looking at the CD66 in these images, we can see high expression of this marker in PDAC or tumor tissues, and very low expression of this marker in other healthy organs. The expression of the CD66 in ovary, here you can see, only occurred in the empty cell mucus region where the antibody binds, and this does not pose a risk of harming healthy tissues. Similarly, CD318 also does not show expression in healthy organs, and this showed no off-target effects for this candidate, and it makes it a promising candidate for further evaluation. On the other hand, when we are looking at TISPEN 8 construct, it showed high level of expression in colon, as you can see here, and this raised some concern about its safety. However, because CD318 and CD66 does not display expression in colon, the TSPN8 can still be used in a combinatorial approach. Overall, the use of Maxima imaging platform helped to ensure that the selected CAR T were tumor specific and safe for further evaluation. In the second part of the study, the selected CAR T were generated from human T cell and then tested first in vitro and then in vivo. And what we see here is the results from in vivo assay. NSG mice xenografted with ASPC1 cell line and treated with CAR T cells of different constructs. So you can see here in the CD66 spacer, we overall see a control over the tumor, but no change in the size of the tumor. TSPNA shows a bit of different results. First, for 2 mice, we see overall control over the tumor and reduction in the size of the tumor for 3 mice. The TSPNA spacer shows overall an effective reduction in the size of the tumor, but the best candidate and the best result is from CD318 with a fast and complete reduction of tumor mass. To further evaluate the CAR Ts, NSG mice were xenografted with another cell line, PDAC cell line called VXPC3. The response to the CD66C, as you can see here, is a slight increase in the size of the tumor mass. Our best candidate, CD318, shows again a fast and complete reduction in the size of the tumor. And then TSPN8 spacer, as we see here, there is a slight reduction in the tumor mass and overall control of it. And then TSPNA-L, we can see that there is only a control with no reduction in the size of the tumor. Here is a flow cytometry analysis of TSPN8 in 2 cell lines that were used in the trials. TSPN8 shows a very low expression in BXPC3 cell line, whereas high expression in ASPC1. And this can be explained why the performance and response to this construct was quite low in the second trial. But we have no explanation of the change in the performance or response to the CD66 in 2 trials. In the previous slide, I showed you the effect of the therapy, which was assessed by tumor size and overall survival of the mice. Here we are looking at an additional analysis of the tumor tissues using Maxima imaging platform. In this image, you are looking at a tumor section, tissue section treated by CAR-T specific for CD318. We see an abundance of CAR-T cells labeled by LNGFR and quite low expression of cytotoxic cells. Here we see a few remain cancer cells, and surprisingly, there is a significant macrophage infiltration, which is a surprise since macrophage is believed to be non-functional in NSG mice. And finally, almost no expression of target in the presence of CD318, and this indicates the successful elimination of tumor cells in early stages. To summarize, Maxima Imaging Platform is the only instrument that can image more than 200 markers on the same sample. It is fully automated and there is no hands-on time required. It has optimized disposables for the analysis of any kind of fixed sample. More than 2,000 pre-tested, ready-to-use antibody conjugated are available. And on top of that, we have a powerful MaxIQ View image analysis software that is specifically tailored for spatial biology analysis. And finally, it's a perfect fit for any type of deep phenotyping, drug discovery, biomarker research, and beyond.
Thank you, Jenny and Sarah, for a great presentation. And we're now going to start our Q&A. Please do keep your questions coming in. We'll get to as many as we can today, and any others we can answer by email. So first up, Daniel, I've got a question here for you. What is the maximum number of markers that you can stain in one sample?
Yeah, so this is actually a very excellent question. And we were also moved by this question in the past. And when we were still in the initial prototype phase, we conducted a test run where we stained 450 different markers on the very same tissue section. And the last 150 markers were the first 150 markers. And so we did this in order that we can nicely compare if we lose quality throughout the run. And in that run, we were actually not able to see any differences in the tissue quality.
Perfect, thank you. And Jenny, a question for you here. Is it possible to design application-specific antibodies that work with the instrument?
Yeah, that's actually also a really good question. So yes, that is possible. So we already from Miltenyi, we have already a very broad portfolio of pre-tested antibodies, over 500 that you can use. But since we cannot have all antibodies in our portfolio, it is also possible to use from third-party platforms antibodies. You can just then conjugate them to fit PE or APC. And so the Maxima is still an open platform where you can also use third-party antibodies. So you can use your application-specific antibody, conjugate it to the fluorochrome, and then use it in the Maxima without any problem.
Great, thank you. And Daniel, coming back to you, does the signal arrays method cause tissue damage?
Excellent question as well, and not as far as we can tell. So we have these three different methods, as explained by Yinya earlier. And a lot of people have actually concerned that either the power of the LED lights is causing some harm to the tissue, or the enzymes that we use for the release of the fluorochromes. And not as far as we can tell, actually.
Great, thank you. And Jenny, I'm back to you for this next question. What is the most important benefit of this multiplex imaging platform when compared to other instruments?
Yeah, that's also a great question. So it's really that we here offer an end-to-end solution, and all from one company and one provider. So it makes you really easy to get fast to your data, since we provide it from the beginning to the end, and we make it really easy for the user to use it. Everybody can use the machine, and yeah, starting with a fully automated instrument, and then over the sample carriers to the broad portfolio and this amazing image analysis. So it's really like a truly end-to-end solution for the spatial biology experiments.
Fantastic, thank you. And staying with you, I have a quick linked question here. Does the analysis software come together with the instrument, and what about updates?
Yeah, exactly. This is part of the imaging platform. So the analysis comes with the instrument and is included in there, and also the updates are including there. So directly, if there's a new update, it pops. There's a pop-up, and then you can update the analysis software directly. So the updates come with it, and of course the analysis software as well. Yes, it's part of the package.
Fantastic.
Thank you.
And one more for you, Daniel. What do you suspect is the potential reason as to why the CD66C CAR is dysfunctional?
Oh, yeah, well, this is a tricky question, and we conducted a lot of research to figure this out. And unfortunately, I have to tell you that we still do not know. However, what I can tell you is that on some point, we just changed the CAR configuration to a shorter spacer, and this one works much better. So we're not exactly sure of the reason. We just think that in this specific spacer configuration, the CAR is not effective enough. And when you use a shorter spacer, it gets better.
Fantastic, thank you. Well, thank you, Jenny and Daniel, for answering those questions. I'm afraid that's all we've got time for today, but any questions we didn't get to, we will reply by email. The webinar will be available on demand from tomorrow, so look out for an email from us with the link. And just as a reminder, if you enjoyed today's webinar, don't forget to join us for part 2, which will be taking place next Wednesday, May the 10th. So all that's left is to thank our speakers once more for some great presentations, and thank you to the audience for listening. We hope you'll join us again soon.
Thank you.
And thanks for having us.
Thank you for the questions.